Hello, today I will be talking about DNA replication. I want to start off by noting that this diagram shows DNA replication after helicase has opened up multiple segments of the double-stranded DNA, and this is known because an origin is indicated, so this is the first place where the helicase would have attached and started to open up the DNA from. We have two helicases attached, one on each strand, because we want to open up the DNA going in both directions in order to make a complete copy of the double-stranded DNA. Helicase is indicated on this diagram by this big filled-in purple circle, and it uses energy from ATP hydrolysis to break the hydrogen bonds between the bases on both strands, and this will open up the double-stranded DNA. After this happens, a single-strand binding protein, multiple actually, are added onto both strands, and this helps to keep the DNA open and not come back together. Then topoisomerase is added, which is indicated by this green, and it's on both ends of the DNA and reduces the torsional stress, which is created by the helicase. It reduces this stress by breaking the phosphodiester bonds on one strand, unwinding it, and then reforming the bonds. After topoisomerase has reduced some of this stress, primase comes in, which is indicated by this not filled in orange circle, and creates the RNA primer, which is about 10 to 12 bases long. RNA is built in the 5-3 direction, same as DNA, and in this diagram, the RNA is indicated by orange, while DNA will eventually be indicated by blue. After this happens, the DNA polymerase III comes in with the sliding clamp to begin DNA synthesis. The sliding clamp keeps DNA polymerase III on the strand. It holds it on there and doesn't allow it to fall off, so it aids in the DNA synthesis. And then the DNA polymerase III will use that primer, hold on to it, that RNA primer, it holds on to it, and it starts to lay down the DNTPs to build the DNA strand. And the first strand built, which is, I'm pointing at right here, is the leading strand. And there's two of those because we want a complete copy of the double-stranded DNA. So we want two double-stranded DNAs at the end of DNA replication. If we only had one leading strand and one lagging strand, so basically DNA synthesis based off of one of the DNA templates and not both, at the end of DNA replication, we would have one DNA template by itself and one double-stranded DNA, which is not the point of DNA replication. We want a complete copy. So this process occurs on both strands of the template DNA, which is why you see two helicases, two primases, two DNA polymerase IIIs, two sliding clamps, two DNA polymerase Is, two ligase, two topoisomerase, etc. So after this leading strand has been built, there's all of this empty space from the helicase. But since DNA replication happens in the 5-3 direction and we can't go backwards, we need to start this process over again for each of the sections that was opened up by the helicase. So the primase comes in, brings in some RNTPs, lays that down for 10 to 12 bases, and then the DNA polymerase III comes in with the sliding clamp, extends DNA until it hits the fragment that was built before it. Normally, there would be some RNA here, but this right here is showing a later step, which I'll get to. But these little fragments that are created are called Ozaki fragments. And there's a problem with them because there's a bunch of RNA that are in these fragments, and we don't want that in our replicated DNA. So we need to remove these and replace it with DNA, which is exactly the job of the DNA polymerase I. This enzyme is indicated by this filled-in orange circle, and it's again one on both strands, and it removes the RNTPs and replaces them with DNTPs. And then there is still an issue after this because, as seen right here, we have the synthesized DNA, which is great, but it's not connected fragments, it's still split up. So we need one additional enzyme, which is ligase, and that's indicated by this not filled in pink square, again on both strands. And it creates a phosphodiester bond between these two DNA fragments. So we have one long continuous piece of DNA at the end of DNA replication, and eventually two double-stranded helixes. Thank you.